Think about the last time a doctor asked for your blood type. Most people answer quickly, almost casually, as if it is just a label, a minor administrative detail sitting somewhere on a medical form between your allergies and your emergency contact. But what if that label was never just a label? What if the 2 letters and 1 symbol that classify your blood were actually a compressed archive of everything? Your ancestors survived? Every battle your immune system won before you were even born, and a biological inheritance so ancient that modern science is only now beginning to decode it properly. That is exactly what is happening. And if you have type O positive blood, the story being uncovered is about you. Let us start with something that almost nobody talks about when they explain blood. Types. Most people know the basic categories: A, B, AB, and O. Most people know the positive and negative designation refers to something called the Rh factor. And most people stop there, assuming the rest is just medical technicality. But here's what the standard explanation always leaves out. Your blood type is not just a classification system invented in a hospital laboratory. The A, B, and O blood types in humans evolved at least 20 million years ago in a common ancestor of humans and other primates. This is not a modern invention. This is ancient biological architecture. And the reason it has survived for 20 million years inside primate DNA without disappearing is one of the most important questions in all of evolutionary biology. Think about what that means for a moment. 20 million years. Civilizations rise and fall in centuries. Ice ages come and go in tens of thousands of years. But this genetic system, the one that determines your blood type, has been running continuously since long before Homo sapiens existed, long before any human ancestor walked upright on this planet. That kind of biological persistence does not happen by accident. That kind of persistence only happens when something is so fundamentally essential to survival that nature refuses to eliminate it. And here is where it starts to get interesting. Type O is considered the oldest blood type, tracing back to our earliest ancestors. Types A and B emerged later, shaped by changing environments and the populations that adapted to them. That is the sequence. O came first. O was the original. What you carry in your body is not just one variation among equals. It is the foundational model from which everything else developed. And the reason that matters is not poetic or philosophical. It is deeply biological. Because being the original means your blood was shaped by the harshest, most unforgiving conditions in all of human evolutionary history. And it survived, every single one of them. But before we go deeper into what that survival actually means for your body today, we need to understand something about how blood types work at the molecular level, because this is where the real story begins. Most explanations stop at the surface. They tell you about antigens and antibodies and compatibility charts, but the actual mechanism underneath those words is far more extraordinary. Your blood cells are coded in proteins. These proteins act as identification flags, biological passwords that your immune system reads constantly, every second of every day, deciding what belongs to your body and what does not. Your body uses these antigen molecules, among their other purposes, as identification markers to separate self from potentially harmful cells. And the configuration of those flags on your blood cells is determined entirely by your DNA, the genetic code you inherited, unbroken from a chain of ancestors stretching back millions of years. What makes type O blood structurally different from all others is what it does not have. Type O blood cells do not carry the A antigen. They do not carry the B antigen. O-type is considered the universal donor because it lacks A or B antigens. The base structure of the system is a specific molecular arrangement known as the H antigen of the O blood group. The H antigen is the foundational layer, the raw original. Surface that type A and type B blood, then modify with additional molecular structures on top. In other words, every other blood type is a modified version of what you already have. Type O is the unmodified original, the template, the base code. Now read that again, because the implications are enormous. Scientists working in laboratories right now are not just studying blood types out of academic curiosity. They are studying them because the molecular architecture of type O blood has opened up entirely new possibilities in medicine. Successful interconversion of blood group types could potentially pave the way for safer and more effective treatment. Opportunities such as faster access to organ and tissue transplants, decreased chances of graft rejection, and reduced risk of transfusion-transmissible diseases. Researchers have been working on enzymatic processes to convert type A blood into type O, essentially trying to recreate what your body already produces naturally. The fact that the scientific community's greatest goal in transfusion medicine right now is to manufacture what type O bodies make automatically should tell you something profound about the value of what you carry. But the story goes much deeper than laboratory medicine, because your blood type does not just exist in isolation inside your veins. It is woven into your immune system, your disease resistance, your cardiovascular profile, and, according to the most recent research, potentially even your neurological function. The connections between blood type and health outcomes have been studied for decades, but the precision with which scientists can now trace those connections has increased dramatically as DNA technology has advanced. And what is emerging from that precision is a picture of type O positive blood that is far more complex and far more significant than the simple compatibility chart most people grew up learning. Consider first what your blood type does for your heart. Type O blood reduces the risk of a heart attack significantly. This protection comes from type O blood carriers having lower levels of procoagulant factor E8 and von Willebrand factor in their plasma. These are clotting proteins. When they are present at high levels, they increase the likelihood of dangerous blood clots forming inside arteries, the kind of clots that cause heart attacks and strokes. Type O individuals naturally produce lower concentrations of these proteins. That is not a lifestyle choice. That is not the result of diet or exercise. That is a genetic inheritance coded directly into your DNA, expressed through your blood type. But here is where it gets more complicated. The same blood type that provides cardiovascular protection does not provide uniform protection against everything. Persons having O-positive blood are susceptible to some diseases like malaria and cholera. They may also have increased risks associated with pancreatic cancer and stomach ulcers. This is not a flaw in the design. This is a feature of evolutionary biology that most people fundamentally misunderstand. No blood type is invincible. No blood type is universally superior. Each type carries a specific profile of strengths and vulnerabilities that was shaped by the particular environmental pressures your ancestors faced. What matters is understanding your specific profile and understanding it accurately using the best available science. What most people are not seeing is this: the vulnerabilities of type O. Blood are not random. They follow a very precise pattern that reveals exactly which kinds of biological threats your ancestors were designed to fight, and which ones they traded away protection against in the process. Type O. Individuals demonstrate remarkable resistance to severe malaria. The parasites that cause this deadly disease have difficulty binding to type O red blood cells, providing natural protection in malaria endemic regions. This protection is so significant that type O blood is more common in populations from areas where malaria has been historically prevalent. This is natural selection in direct action visible in living human populations today. In regions where malaria was historically the primary killer, large parts of Africa, South Asia, and Southeast Asia, type O blood became genetically dominant because the people who carried it were more likely to survive long enough to have children. Your blood type is literally the biological receipt from a survival transaction your ancestors made thousands of years ago. And here is the part that connects all of this to cutting-edge DNA science. For most of human history, scientists could only study blood types from living people. They could observe patterns in populations, note correlations with disease, and make inferences about evolutionary history, but they could not actually look back in time. They could not examine the blood type genes of ancient humans. They could not trace exactly how and when these genetic variants spread across the planet. That limitation has now been shattered. Scientists have been able to study previously sequenced genomes from 22 Homo sapiens individuals who lived from 46,000 to 6,500 years ago, 14 Neanderthals who lived roughly between 120,000 and 40,000 years ago, and a single individual from about 98,000 years ago, thought to be descended from both Neanderthals and another close cousin, the Denisovans. This is not hypothetical reconstruction. This is actual ancient DNA analysis. The genetic code that determines blood type has been extracted from bones tens of thousands of years old. And the story it tells is extraordinary. What the ancient DNA reveals is that blood type evolution was not a slow, gradual process happening randomly across all human populations. It was targeted. It was directional. It responded dynamically to specific environmental pressures as human beings moved into new territories. As Homo sapiens migrated out of Africa between 70,000 and 45,000 years ago and expanded into Eurasia, new genetic variants determining blood groups emerged and entrenched themselves in these roving populations. Every single ancient human they looked at had unique variants. It was very unexpected that they would have evolved in such a short time. Think about that. Every single individual examined had unique blood group variants emerging rapidly in evolutionary terms as people moved through different environments, encountered different pathogens, and faced different survival pressures. The blood type system was not fixed. It was actively adapting generation after generation, responding to the world that humans were moving through. And the Persian Plateau connection is one of the most striking findings in recent ancient DNA research. Archaeological evidence suggests that along their journey out of Africa, Homo sapiens groups paused and remained in the Persian Plateau, a region between the Zagros Mountains and Iran, for about 15,000 years. Researchers believe that would have been enough time for humans to develop the genetic variation underlying new blood types only found outside of Africa, a sort of genetic incubator period before these humans continued their migration. A genetic incubator, a 15,000-year laboratory where human DNA was being tested against new environments, new pathogens, new climate conditions, and the genetic configurations that helped people survive were selected, reinforced, and passed forward. The blood that flows through your veins today was in part forged in that crucible. This is not abstract history. This has direct implications for understanding your biology right now because the genetic variants that were selected in those ancient populations are still operating in your body. The same molecular mechanisms that helped your ancestors survive plague and famine and epidemics are still running in your immune system, still regulating your cardiovascular function, and still shaping how your body responds to stress and infection. According to informed sources in the scientific community, the blood type research that has emerged in 2024 and 2025 represents a genuine inflection point in our understanding of human genetic heritage. Researchers from the UK and Israel described an entirely new blood group system in humans in 2024, the result of nearly 20 years of investigation into unexplained immunological anomalies. And this is only the beginning. As of 2024, the International Society of Blood Transfusion recognized 45 blood group systems. A newly discovered mutation in a gene called PIZ, which alters how proteins anchor to the surface of blood cells, led to the recognition of a 48th blood group system in 2025. The science of blood typing is not finished. It is accelerating, and every new discovery reshapes what we understand about the ancient genetic systems that include type O positive. Now let us talk about something that the mainstream conversation around blood types almost never addresses, the COVID-19 connection, and what it revealed about type O blood's immune architecture. Because the global pandemic of 2020 became unintentionally one of the largest natural experiments in blood type immunology ever conducted. Billions of people were exposed to the same pathogen across hundreds of countries, with decades of blood type data available for comparison. What the data showed was significant. Several studies suggest that people with blood type O and people who are Rh negative have less chance of testing positive for COVID-19, and the mechanism behind that protection is particularly revealing. Scientists believe part of the correlation between blood type and COVID is how well your blood type helps you fight off inflammation and infection. Research found that people with type A blood have higher levels of von Willebrand factor naturally than people with type O blood. When the COVID virus damaged the lining of blood vessels, the resulting clot response was more dangerous in people with high VWF levels, contributing to strokes, kidney failure, and pulmonary embolisms. Type O individuals, with their naturally lower VWF levels, were partially buffered against the most catastrophic vascular complications of the disease. This was not predicted in advance. It was discovered through observation of millions of real-world cases, and it added another chapter to the already extraordinary story of how type O blood's molecular architecture provides advantages that were coded into human DNA long before the pathogen it was protecting against even existed. This brings us to one of the most philosophically striking aspects of blood type science. Your immune system does not learn, it inherits. The protections you carry were not developed during your lifetime through exposure and adaptation. They were preloaded into your biology at the moment of conception, encoded in the DNA that combined from your parents, which was inherited from their parents, stretching back through an unbroken chain of survivors all the way to the earliest members of our species. Every piece of that inherited protection represents a battle that was fought and won by someone who came before you. Every molecular advantage in your blood type is a biological trophy from a survival victory in your ancestral past. And the numbers that surround type O positive blood make this even more striking when you examine them properly. More than a third of the world's population shares type O positive blood, and nearly 100% of the population of Central and South America has O positive blood. Think about what that geographic concentration means in evolutionary terms. The populations of Central and South America descend from ancestors who crossed from Asia into the Americas during the last ice age, approximately 15,000 to 20,000 years ago, through a land bridge across what is now the Bering Strait. These were small populations, moving through extreme environments, facing severe cold, unfamiliar pathogens, and profound isolation from the rest of humanity. And they arrived in the Americas carrying almost universally type O blood. Whatever genetic selection process had been operating during those thousands of years of migration had converged almost completely on a single blood type. That is not coincidence. That is the fingerprint of survival pressure operating at the genetic level. O positive blood is found in about 37, 4% of people globally, and can be given to all Rh positive blood types, including A+, B+, AB+, and O+, making it vital in emergency care. But here is the thing that hospitals never have enough of. Type O is routinely in short supply and in high demand by hospitals, both because it is the most common blood type and because type O negative blood is the universal blood type needed for emergency transfusions and for immune deficient infants. There is a paradox embedded in that statistic. The most common blood type is also perpetually the most needed, because when a patient arrives in an emergency room and there is no time to type their blood, when someone is bleeding and every second counts, the blood that saves their life is type O. Hospitals consume it faster than it can be donated. The demand is constant. The supply is never sufficient. What most people are not seeing is the larger meaning of that medical dependency. It is not just logistical. It is historical. Hospitals running on type O blood in emergency situations are, in a very literal sense, relying on the oldest immune architecture in human evolution to keep people alive. While more precise matching is determined, the blood type that your ancestors carried through every major crisis in human prehistory is still the biological safety net of modern medicine. The oldest solution is still the most reliable one. Now let us talk about the future of this science, because this is where things become genuinely uncertain and genuinely consequential. The enzymatic research currently being conducted on blood type conversion is moving fast. Certain glycoside hydrolases are capable of cleaving the terminal monosaccharide that distinguishes type A blood from type O blood, raising the possibility of converting type A blood to universal type O blood by enzymatic cleavage. If this research succeeds at clinical scale, the global blood supply could be fundamentally transformed. The shortage that hospitals face and the perpetual life-threatening deficit of type O blood could theoretically be resolved by converting other blood types into it. But notice what that means. It means converting blood to resemble type O, not converting it to type A or B or AB, converting it toward the original, toward the oldest, toward the foundational template that evolution produced first, and that DNA science is now trying to recreate artificially. There is something almost philosophical in that. For all the advancement of molecular biology, for all the gene editing technologies and CRISPR breakthroughs and next-generation sequencing platforms, the target that medical science is aiming at, the destination it is trying to reach through artificial means, is the blood configuration that nature already produced in the bodies of people who lived before recorded history. But the research does not stop at the molecular level. The broader genomic studies that are emerging from ancient DNA analysis are reshaping the entire framework through which scientists understand human biological diversity. The biggest ever study of ancient human DNA, published in 2026, shows that human evolution has actually accelerated over the past 10,000 years, with data from more than 15,000 ancient people, revealing natural selection of hundreds of genes linked to immunity, skin tone, behavior, and other traits. 10,000 years. That is not the deep evolutionary past. That is recent. That is the period during which agriculture emerged, cities were built, populations exploded, and pathogens evolved in response to human density. The immune systems being selected during that period, including the blood-type variants being reinforced or diminished, were responding in real time to the new disease landscape that human civilization was creating. And you are the living result of that selection process. The new blood type discoveries that have emerged in the last 2 years add further layers to this picture. The male blood group system established through research published in the journal Blood was the result of a collaborative team effort involving scientists from multiple institutions. The protein involved, MAL, is very small, with some interesting properties that made it particularly difficult to identify, requiring multiple lines of investigation to accumulate sufficient proof. There are currently 48 recognized blood group systems. Each system represents a different set of proteins on the surface of blood cells. Each set of proteins has its own evolutionary history, its own pattern of genetic inheritance, and its own implications for immune function and disease susceptibility. What we know about blood types in 2025 is vastly more sophisticated than what was known a decade ago. And what will be known a decade from now will be vastly more sophisticated again. For people carrying type O positive blood, this accelerating science carries a specific significance, because type O is not just the most common, it is the most studied, it is the most ancient, and it is the blood type against which all other variants are in some sense measured. Because it is the original configuration from which the others diverged, every new discovery in blood type genetics adds another dimension to the portrait of what type O blood actually is at the molecular level, and what carrying it means for the body that contains it. The story of the evolution of blood types is an intriguing one, full of the history of human adaptation. As ancient Homo sapiens left Africa to enter a whole new world of different environments, their blood types adapted to new challenges. These genetic changes reflect the resilience and ingenuity of our species, as well as the lasting impact of interactions with Neanderthals. The more genetic secrets of our ancestors are unveiled, the more one can appreciate the complex interplay of forces at work as humanity made its journey across the globe. That journey, from the first modern humans leaving Africa through the genetic incubator of the Persian Plateau, across the Ice Age corridors of Central Asia, all the way to the final isolation of populations in the Americas, is written in the blood type distribution of the human species today. You can read the map of ancient human migration in the frequency of blood types across different populations. And everywhere you look, type O is the common thread, the oldest signal, the most persistent pattern, the biological signature of the very first chapters of the human story. What does all of this mean for you practically today? It means that your blood type is worth understanding at a level far beyond blood bank compatibility. It means that the health correlations, the cardiovascular advantages, the specific infection resistances, and the particular vulnerabilities are not folklore or pseudoscience. They are the measurable biological consequences of millions of years of evolutionary pressure, now readable in precise molecular detail through modern DNA technology. It means that as personalized medicine continues to advance, as genetic sequencing becomes cheaper, faster, and more clinically integrated, your blood type will increasingly be considered not as a standalone marker, but as one layer within a much richer picture of your genomic inheritance. And it means something else, something that the pure clinical data does not quite capture. You're not the first person to carry this blood. You're the latest in an unbroken line of carriers, stretching back. Further than any human record, further than any civilization, further back than language itself. Every person who ever shared your blood type and survived a plague, a drought, a war, or a pandemic, they passed something forward. Not a memory, not a story, but a molecular configuration, a biological architecture that kept them alive long enough to reproduce, that now runs silently inside. You, doing the same work it has always done, largely without your awareness. The science is accelerating. The discoveries are coming faster. Every year, the picture of what blood type actually means at the genetic level becomes more detailed, more precise, and more consequential. What researchers find in the next decade will almost certainly include discoveries that change the way medicine approaches blood type in clinical practice, moving beyond simple transfusion compatibility toward a genuine understanding of how blood type intersects with immune function, disease risk, drug response, and possibly even cognitive biology. But for now, what the DNA has already revealed is this. If you have type O positive blood, you're not simply carrying a common biological label. You are carrying the oldest surviving immune architecture in human evolutionary history. You're carrying a molecular record of every critical survival pressure that shaped the Homo sapiens lineage from its earliest days on this planet. You are carrying the blood that nature built first, tested longest, and preserved most widely, the original code that everything else was written from. The question that science has not yet fully answered and may not answer for years is this: what else is written in that code that we have not yet learned to read? What other biological advantages? What other hidden resistances? What other deep evolutionary gifts are encoded in the structure of type O blood that current technology cannot yet detect? The researchers who are working with ancient DNA, enzymatic conversion, and next-generation blood group genotyping are pulling back the edges of something enormous. They can feel the outline of it, but the full picture is still forming. And that is precisely why this story is not finished. The blood running through you is older than history.